Hello everyone, how are you doing? Today I'm here to talk you about a little bit about my profession. I'm a scientist. And no, it's not like the movies. I'm a professor at the university and I'm a researcher too. To get there, I get a bachelor's degree in biology and after that a master's degree and a doctorate in genetics. And today I teach classes and work in research. The focus of my research is chromosomes, which is where genetic information is stored. You have 23 pairs of chromosomes, a half from your dad and a half from your mother. Which is why you have some traits from one and others from the other one. We can take a look at the chromosomes under a microscope and figure out there is a connection to genetic diseases. That's what we'll do here. And today I will show you a little bit of my routine at the lab and I also introduce you to other researchers. Are you ready to see my workplace? Let's go! This is the entrance to the building where our lab is located at the State University of Maringá. This is the Department of Biotechnology, Genetics and Cell Biology that I have with the different labs and classrooms. Now we arrive at our lab. This is the teachers and researchers room. And now, this is the main room, when the equipment and reagents are kept, and also students conduct research. And meet Mazé, our lab technician.
Hi there, this is our lab. Here we studied the DNA. This is the DNA, the smallest molecule in our cells. This DNA is organized in chromosomes like our mascot. This is Dr. Genetics. And the cells can be shown at the cells and we can observe it in a microscope. Here you can see 23 chromosomes, 23 pairs, a half on your mom and a half on your dad. And this is the small cells. We analyzed and organized them in a karyotype for looking for some problems or diseases like Down syndrome or another. A little bit from our research, but there are many researchers in the university. You will see the other videos from other labs. Take with us.
So let's understand how this experiment works. First, we choose the target pollutant to be studied, for example, a pollutant called X. Then we can put it in contact with the fish in different percentages, such as 25 or 50%, to evaluate the activity of the compound. In this experiment, at the same time, there will be fish living in clean water and others in contaminated water, as in tanks B and C. After a period of exposure, for example 1 month, the fish are then sacrificed to analyze their blood and organs. At this part, it's important to notice that the fish don't suffer. The entire protocol of the Animal Research Ethics Committee is followed, and the animals are anesthetized before euthanasia. Furthermore, the cells are prepared and transferred to glass slides so that they can be analyzed under microscope. And in the end, we have our cells slides that can be seen under the microscope. We can see their nucleus in a pink and purple color. We can also see their chromosomes, and all of it will depend on how we manage our cells until the final results. And looking to the cells through the microscope, we can see certain type of alterations, and we can compare the cellular changes that occurred in the fish due to the action of the pollutant in different concentrations. And by evaluating the final data of our analysis, we can establish safe exposure doses for the environment. And this is how Professor Eliane's research takes place in the Animal Cytogenetics and Mutagenesis Laboratory, but other types are also performed in our department. So, hello everyone, my name is Myllena, and I'm currently a master's student associated with the Environmental Biotechnology program at the same university, and I'm here to show you other types of research areas that you can pursue that may be associated with genetics. So, firstly, we have the Biological Control and Insect Bioprospecting Laboratory. Here, researchers can study the pests that run agricultural crops and those that can be even associated with forensics. And continuing in this direction of the insect studies, they can also research the ones that can benefit ecology, medicine, and pharmacy. So, another one is the Microbial Biotechnology Laboratory, where they study different types of microorganisms, such as endophytes, those that can be found inside plants and don't cause damage to it. Other studies are related to the selection of agents for bioremediation, which means that these organisms can help in the degradation of molecules that are difficult to clean from the environment. And they can also work in obtaining nanobiocompounds and also in the selection of agents that are resistant to radiation and genetically modified organisms. In addition, we have a laboratory related to animal genetics and biotechnology, in which we have insects such as bees being used as instruments for toxicology to understand their resistance to insecticides and even their genetic diversity. They can also be used to study gene expression and the selection of queen bees for honey production. The Plant Genetics and Biotechnology Laboratory works with the cell culture of plant tissues, such as those from the cereals pyrovianus, and with their production of metabolites. In addition, there are also studies on the productions of soma clones from the species and the genetic diversity promoted by the in vitro culture of different tissues. And last but not least, we have the Environmental Monitoring and Mutagenesis Laboratory, in which I participated, where we research basically the toxic genetic effects of different molecules and drugs on cells and other organisms, such as mice or even the cell roots of allium cepa. I would like to pay a little more attention to the animal cell culture, where we take cells considered normal or tumorals from different animals, such as mice or even humans, and test them in a controlled space to assess the toxic effect of a certain compound. After the culture, we can see different types of DNA damage through some tests such as the COMET assay or the MTT, and through this type of study, which is considered across the world to approve initial research on living beings, we can begin to understand whether a compound may be beneficial or not for later use. So, These are some of the areas of research you can work with in the biotechnology, and I'm sure there's a lot more to be explored in other fields. So keep searching, and I'm sure you'll find something that might spark your interest in being a scientist in the future. Just keep in mind that research work is very detailed, and the science needs to be dedicated and studied constantly.